Hi, my name is Jan Klaassen. I'm a neurocritical care physician at Columbia University. I had the honor of chairing the working group on for common data elements, focusing on recommendations from the Electrophysiological Working Group. The focus here is to advance coma science and to provide some guidance on the acquisition and analysis of electrophysiological tests. The idea is to provide a nomenclature for naming, defining, and structuring data for clinical research variables, which was currently lacking. So the idea was to put a multidisciplinary international group together, and we were able to involve people with different backgrounds. So there were clinician scientists in the group that had a focus in the more acute care setting. There were people that were focusing more on the chronic disorders of consciousness. There were people with expertise in pediatric neurocritical care represented in the group. There were several scientists, pure scientists involved. We also had a pharmacist join the team and also a trainee in neurocritical care who ended up being the first author of the manuscript that we put together. So first, we actually looked through existing common data elements. We looked at other common data elements that had been developed for diseases like stroke or epilepsy, and looked what we could use from that. And then went through all of them and then identified areas that were missing and developed and proposed common data elements in iterative processes with multiple meetings to sort of develop common data elements for these areas that were not covered yet. So electrophysiology encompasses a number of different tests that we use in clinical practice and in research to assess the brain. This includes, for example, EEG, which is just recording the electrical activity of the brain. It includes evoked potentials, for example, where a stimulus is provided to a peripheral nerve and then the response is recorded from the brain. It involves also active tests where patients are asked to participate in a task and then the EEG is recorded and analyzed. These tests EEG has been around for many, many years, now over 100 years. But what has really made it possible to use it in the acute care setting is the development of computers. Being able to record the EEG digitally and then process it afterwards has allowed us to use it in, I want to say, electrophysiology unfriendly environments like the ICU, where there's a lot of artifact around, a lot of confounders, and that allows us to post-process the EEG and get very useful and important data out of it. There's an enormous body of research that has supported these techniques, but it was time to develop a common, I want to say the common data elements are almost like a common language that we use so that we call things by the same name if we talk about the same thing. And so this is sort of a fundamentally important step in advancing science and has been identified by the NIH many, many years ago. And we wanted to provide a tool set for that for disorders of consciousness. So I think one important thing is that together with the digitally recorded EEG, it's also important to acquire some additional information, not just the EEG itself. So it's important, one, sort of to acquire what kind of technical context was used to record the EEG. For example, what is the sampling rate that the EEG was recorded with? What was the impedance? Things like that are crucially important to interpret the EEG data afterwards. I would also recommend collecting some information about patient behavior. Ideally, you use like a highly standardized test like the Coma Recovery Scale Revised, but record some patient behavior. That's very, very crucial. Afterwards, sort of trying to put it together from from recorded patient records is very inaccurate and not very useful. And then also to get some information about confounders that may have been present. One example would be, for example, the sedation. Record what kind of sedation the patient was on, what type of sedation, what doses the patient was on. So these are fundamentally important to understand when these CDEs are being used in practice and have been developed by the NIH. So there are CDEs that fall in the category of general core CDEs. These are required for all studies, regardless of the neurological disease. They cross diseases, so you may find them actually in stroke or in epilepsy, but also in disorders of consciousness. Then there are the CDEs that fall into the category of disease core. These are required for all disorders of consciousness studies, regardless of the neurological disease that is causing the disorder of consciousness. The next category is basic CDEs. These are also known as supplementary or highly recommended CDEs. They're strongly recommended for all DOC studies. The fourth category is supplemental, and these are recommended for specific DOC studies, depending on the context and the goals of the study. The exploratory CDEs are the ones that can be considered for use in DOC studies, but require further validation. And then very, very important, and this you will find in the electrophysiology CDEs quite prominently, but also in others, are key design elements. And these are these sort of contextual information about, like, for example, the sampling rate that was used to acquire the electrophysiological data. You can also think of them as this methodological parameters that are used. So I think it's sort of an exciting time to go into this research. There's exciting work developing using the CDEs already ongoing. For example, for EEG, there's a large number of resting EEG studies that are used to sort of study patients that have an acute disorder of consciousness. Some of the computation analysis that is applied now allows us to sort of identify very, very important information like connectivity in the brain using these EEG studies. Another example are studies that look at sleep. So there's a number of important parameters that you can derive from just resting EEG. Then there's a category of, I want to call them evoked responses using EEG. That allows you, for example, to present the patient with a task, like asking the patient to do something, and then analyzing the EEG and seeing whether there was a response to it. This can, for example, be used to identify cognitive motor dissociation or covert consciousness. And lastly, there's also TMS EEG, where the patient is sort of exposed to a magnetic stimulus, and then the EEG is recorded and the connectivity in the brain is analyzed. So there's a lot of exciting work ongoing already and starting that utilizes EEG and the EEG CDEs. So these can be acquired downloaded from the Zenodo website, and I think you will find this link here. The Zenodo website allows you to download them for free. There have been already 3,000 downloads as of today, so they're very, very popular. But I highly recommend looking at them if you are thinking about starting a study that you're planning. So I think we had an exciting point with our work. Having agreed on a common language, which is the CDEs, allows us to investigate mechanisms of coma and also develop treatments to support the recovery. I just want to urge you all to collaborate together across the world on coma science and advance this field together. Thank you very much.